Good evening to everyone. I am Dr. P. Uma Maheshwari, Associate Professor and Head, Department of Botany, Sri Sharada College for Women, Autonomous, Salem 16. Today we are going to see an overview of plant breeding. Plant breeding. It is a production of new crop varieties which are superior to their parents. It can be evolved by means of selection, introduction, hybridization, through ploidy, mutation and tissue culture. Definition for plant breeding. Plant breeding is defined as changing the biological makeup of plants and crops to increase their yield and usefulness using various plant breeding methods. Or it can be described as an art and science of changing the traits of plants in order to produce desired characteristics. Or otherwise, plant breeding is the genetic improvement of plants for human benefits. Or otherwise, plant breeding is a science based on the principles of genetics and cytogenetics. It aims at improving the genetic makeup of the crop plants. It's the ability of breeder to make a distinction between desirable and undesirable character, that is shattering versus non-shattering pods or spike, more tillering versus low tillering. The selection skill does not have any underlying scientific facts. The science part of plant breeding is with the advent of inheritance laws and the genetic role, it become easy to transfer desirable traits through crossing methodology or through biotechnological approach, mutation, polyploidy, etc. Plant breeding aims to improve the various characteristics of plants so that they become more desirable, agronomical and economical. Thus, the chief objective of plant breeding is to develop such improved varieties of the crop plants that will commercially successful. Generally, a successful variety is one with a total balance of traits that makes it more profitable for the growers than any other one they might choose. Therefore, specific objectives would vary greatly depending on the crop and region. Some of the main objectives of plant breeding may be summarized here. First one is to produce higher yield. Next one, resistance to disease and pests. Then change in maturity period. Then elimination of toxic substances. Then synchronous maturity. Then photosensitivity. Then resistance to abiotic stress. Then improved quality. So these are the various objectives of plant breeding. First one, we will see about the high yields. Most of the breeding programs aim at higher crop yields. In fact, higher yields will always remain one of the main objectives of the breeding program. This is achieved by developing more efficient genotypes, that is, example, hybrid varieties. Next comes improved quality. To improve the quality of the grains that plant produce determines its suitability for various uses. Therefore, quality is an important aspect for plant breeders. Example, milling and baking quality in wheat and cooking quality in rice. Next come disease and insect resistance. Resistant varieties offer the cheapest, the most convenient and environmentally safe method of disease and insect management. In some cases, they offer the only feasible means of control. Example, rusts in wheat. Resistant varieties not only increase production but also stabilize it. Next come change in maturity duration. Breeding for early maturing crop varieties or varieties suitable for different dates of planting may be important objectives in many crops. This would permit a new crop rotations and often extend the crop area. For example, the development of wheat varieties suitable for late planting has permitted in rice wheat rotation. Agronomic characteristics. Modifications of agronomic characteristics such as plant height, tillering, branching, erect or trailing habit, etc. is often desirable. For example, dwarfness in cereals is generally associated with the lodging resistance and fertilizer responsiveness. Then photo and thermosensitivity. Development of photo-insensitive and thermo-insensitive wheat and photo-insensitive rice varieties has permitted their cultivation in new areas. Rice is now cultivated in Punjab while wheat is major rabi crop in West Bengal. In case of wheat, photoperiod insensitivity is due to genes PPD1 and PPD2 which show polymeric gene action. Synchronous maturity. It is very important for the crops. Synchronous maturity is highly desirable crops like moong where several pickings are necessary. Non-shattering characteristics. This feature would be of great value in a crop like moong where shattering is a major problem in case of many commercial varieties. Determinate growth. It is also important. Development of varieties with determinate growth habit is desirable in crops like moong, PGNP, etc. Dormancy. In some crops, seeds germinate even before harvesting. If there are rains at the time of maturity, example, moong, barley, etc., a period of dormancy in such crops would check the losses due to pre-harvest rains. But in some crops, it may be desirable to remove dormancy. Moisture, stress and salt tolerance. Development of varieties for rained areas and for saline soils would be helpful in increasing the crop production in India. The major proportion of calcium up to 70% of cropped area and there are 7 to 20 million hectares are salt affected soils. Elimination of toxins. Some crops have the toxic substances which must be eliminated to make them safe for the consumption. For example, Lathyrus sativus seeds have a neurotoxins that is beta-N-oxalate, beta-dimorphopropionic acid that causes paralysis of lower limbs. Similarly, brassica oil has erucic acid which is harmful to human health. Removal of such toxic substances would increase the nutritional value of these crops. Others. Traditionally, maize is a kharif crop, but scientists are now able to grow maize as rabi and zaid crops. The main steps in plant breeding are given below. The following are the main steps to do the plant breeding work. First one, domestication, through which the creation of genetic variation. The genetic variation may also brought through mutation. In domestication, next comes plant introduction. After plant introduction, hybridization techniques, then germplasm collection. Genetic variation leads to selection of plants, evaluation and release as a variety, then seed multiplication and distribution. Again, the creation of genetic variation also through mutation leads to polyploidy, tissue culture and genetic engineering. We will see one by one. What is introduction of plants? It is nothing but to introduce the various crops or the high yielding varieties or the disease resistant varieties from one area to another area. So it is called introduction of plants, that is process plants introduced from their native place to another place for crop improvement. In that, after introducing, the acclimatization process is very important. It is nothing but the plant can accommodate that environmental conditions and their growth and the yield. So introduction of a plant from their native place to another place which having different climate. Sexually reproducing plants introduced by means of seeds. Vegetatively reproducing plants introduced by means of cutting. So these through seeds, through cuttings. Then acclimatization. It's for successful introduction. The introduced plant has to adapt itself to a new area. Ability of introduced plant to survive in the new climate and soil condition. While introducing the plants, we have to see whether the plant is in healthy nature. Otherwise, if it introduced also with that disease, then that also affect the plants. So the important concepts in plant breeding. Plant introduction and acclimatization already we have seen. Then through pure line selection, mass selection, pedigree method, bulk method, single seed descent method, backcross method and heterosis breeding. The special concepts are mutation breeding, polyploidy breeding, transgenic breeding and molecular breeding. We will see one by one. Pure line refers to the homogeneous progeny of self-pollinated homozygous plant, that is pure line breeding. If the crossed plants are homozygous, then it will pure for its character. Then mass selection. It is a method of breeding in which individual plants are selected on the basis of phenotype from a mixed population. Their seeds are bulked and used to grow the next generation. The deliberate mixture of isolines, closely related lines or unrelated lines are referred to as multilines, and the method of developing a variety from any of these mixtures is known as multiline breeding. Breeding methods. First one, inbreeding, outbreeding and heterosis. What is inbreeding? That is mating of parents who are closely genetically related with each other. Results in increased homozygosity, which can increase the chance of offspring become the similarity from the recessive traits. So that is inbreeding. What is outbreeding? Mating of unrelated individuals, also known as crossbreeding. The offspring formed by mating of two unrelated parents. Types of outbreeding. Intraspecific. What is intra? That is between. So mating between or cross between the members of same species. For example, the lady's finger species, they can cross with each other. That is that between two varieties, if the crossing is made, then it is called intraspecific. Then interspecific, mating between members of different species. That is within the genus, two species can be crossed. So that is called interspecific, that is in between two species. Then intergeneric, mating between the members of different genera which belongs to the same family. For example, Solanum nigrum, Solanum melangina, Solanum tuberosum, Solanum suratensis. Like that, within the genus, the cross can be made. In the family between two different genera, if the cross is made, then it is called intergeneric. So these are some examples for types of outbreeding. If this is interspecific cross, this is intergeneric cross. That is tomato and potato between two genus. That is the genus, the genus between two species within the genus. Solanum tuberosum and Solanum persicum. The next one is heterosis breeding. The increased growth vigor or yield of hybrids over the parents are known as heterosis or hybrid vigor. That is, the plant is in abnormal phenotype or abnormal growth, then it is called heterosis. So it is very much useful in the leafy vegetables, that is for greens. Through this method, if we get large sized greens, then it will be very much useful. Crop breeding to manifest heterosis called heterosis breeding. It brings out the superiority in F1 individuals, but the vigor tends to decrease from F2 generation onwards. What is heterosis breeding? Heterosis means deviation of offspring from the actual character of plants. So in plants, heterosis appears due to developmental stimulation induced by the union of gametes coming from two genetically complementing parents. Theories of heterosis. Two popular hypotheses to explain heterosis in F1. One is dominance hypothesis and another one is over-dominance hypothesis. Method for heterosis breeding. Methodology of heterosis breeding varies with different crops. Depending on their nature of pollination, sexual incompatibility and other reasons, it includes mainly three types: producing inbred lines, testing of combining ability of inbred lines, production of hybrid seeds. Next one, the germplasm. It is the sum total of the hereditary material, that is all alleles of various genes present in a crop species and its wild relatives are called germplasm. A germplasm collection of a crop species consists of large number of lines, varieties and related wild species of the crop. Such collection are also called gene banks. NBPGR, New Delhi has the Indian National Gene Bank and maintains germplasm as seed banks, field banks, slow growth cultures and cryopreserved accessions. Plant breeding technologies and crop improvement. Germplasm improvement in higher yields starts with open pollinated selection, then hybridization through pedigree breeding, statistics, quantitative genetics, plot mechanization, computer technologies, winter nurseries, biotech genetic engineering, transgenic traits, analytical technologies, genomics, proteomics, and finally bioinformatics. What is the role of plant breeding in human welfare? Human beings are dependent on the plants for food. Breeding of field crops provides us food either directly, that is through food grains, or indirectly through meat and milk. Then comes shelter. In addition to food, by produce of agriculture farms are used in making shelter by farmers of rural areas. Then next comes clothing. Clothing for fiber crops like cotton provides clothes for the human population. Fuels. Crops like euphorbia and jatropha are used for biofuel production. Breeding of such crops tackles the problems of energy production for rapidly increasing human population. Nowadays, maize is also used as an important source of ethanol production. The important one is drugs. Breeding of medicinal plants plays an important role in production of many important drugs. These drugs are used for treatment of various human and animal diseases. Entertainment. Flowers play an essential role in people's celebrations and everyday lives like weddings, Christmas, etc. Most of the medicinal plants are seasonal in nature. Shifting the seasonal timing of reproduction is a major goal of plant breeding efforts to produce novel varieties. What are the advantages and disadvantages? In many crops, F1 hybrids are early in maturity. Example, cabbage, onion, tomato, etc. They produce goods with uniform size. Again, example, onion and cabbage. They are resistant to biotic and abiotic stresses. They are always high-yielding varieties. And some disadvantages are production cost is so high. Fresh seeds is to be purchased every time to raise new crop. Sometimes F1 hybrids are vulnerable to disease. Importance of plant breeding. Increases in yield are derived both from improved varieties and from improved management. It suggests about a 50-50 of crop yield split between genetic gain and gain attributed to management. Planting material is the most important input in agriculture. An improved cultivar is the most economic and least laborious input for crop cultivation. Challenges of plant breeding early in 21st century. Human population growth. Human population is growing faster than increases in food production. Grain production increases around 0.5% a year, while human population growth is about 1.5% a year. Next comes sustainability. Environment problems due to natural resource over exploitation. Globalization. Worldwide competition among the largest multinational as global business entity and national companies of agriculture input. Production such as seed materials. Global warming. Changing climate may change in quality and quantity of biotic and abiotic factors. So now we will see the milestones in plant breeding. The first comes 10,000 BC, the domestication of wheat. Crop domestication. During those times, farmers select the best species to create crops. Then, plant breeding based on cross breeding. In these 1926 in that time, development of improved varieties by combining good characteristics from their parents. Again, in 10,000 BC, domestication of wheat, Mendel's laws. Brigadier John Mendel described the inheritance of traits from one generation to the next. His laws become the core of classical genetics. And for the 10,000 years, farmers and breeders have been developing and improving crops. Then these years, 1940s, blast-resistant rice. That is through mutagenesis, developing new genetic diversity by exposing crop plants to chemical agents for radiation. And those times in 1926, more vigorous hybrid corn produced, crossing two genetically different individuals to develop better performing hybrids. So for 150 years, plant scientists and breeders have improved plant breeding on a scientific basis. Today, farmers feed at least 10 times more people using almost same amount of lands as 100 years ago through plant breeding based on genetic information, that is development of improved varieties by working directly with DNA and understanding the structure of DNA and genetically modified organisms, that is introducing foreign genes into the DNA of a plant. Then in 2000, Marker assisted selection has been made. And now targeted breeding using modern tools such as genome editing for more targeted breeding. More desirable characters can be incorporated and through which the new varieties can be produced. In the near future, by 2050, we will need 50% of more food to feed the rapid growing population. Hope clearly you will all understand an overview of the plant breeding. So what are the things, what is plant, we have seen in the last 25 minutes. What is plant breeding? What are the objectives of the plant breeding? Aims and principles of plant breeding. What are the methods used in plant breeding to bring the betterment of the crops? And here I have shared the quiz questions for the test, and I have given the study material in this link. Hope you will all understand this topic in better way. So you think so many subjects involved in this plant breeding. So hope you will all understand this concept. Thank you one and all.